Yeah, my name's Dean Clift. I'm a postdoc at the LMB in Cambridge, which is a medical research council laboratory. Yeah, so we're working on a few different things in the lab. So mainly it's mechanisms of intracellular immunity, so how invading pathogens are intercepted in the cytosol once they've already infected cells. And in particular, we're interested in one protein called TRIM21, which is an antibody receptor. So it binds to the FC region of antibodies, but it's expressed in the cytosol. And this is kind of unusual because we think of antibodies acting outside the cell, right? But it turns out what TRIM21 does, it's in the cytosol waiting for invading pathogens that infect cells with antibodies bound already to their surface. So what happens when these invading pathogens that are coated in antibodies enter the cell, they're then detected by TRIM21, which binds to the antibodies that coat these pathogens. And this protein is quite interesting because it's also a ubiquitin ligase. So what it does once it binds to these antibody-coated pathogens, it catalyzes ubiquitination, which then recruits the proteasome to then degrade these pathogens that enter the cytosol, thereby destroying them so they can't infect the cell. So that's our main interest in understanding the mechanisms of how this is happening and how this translates into the situation in our body. So what's TRIM21's function in response to infection in the body. And then on the other side of that, what we've done more recently is we've adapted this natural function of TRIM21, which is to bind antibody-coated targets and cause their degradation. We've adapted this as a protein knockdown tool, which we've called TrimAway. And so the idea behind this is that you can just take any off-the-shelf antibody that you use for all other applications, and then you can, if you deliver this antibody inside cells, where it binds to an intracellular target of interest, this could be any cellular protein, then if you also have TRIM21 inside these cells, either endogenously expressed or overexpressed, then what happens, the antibody binds to its target, TRIM21 then comes and binds the antibody and causes degradation of that target. And we've shown you can use this to knock down rapidly any intracellular protein. So you can degrade this protein with a half-life of 9 minutes. And then many labs around the world now are using this TrimAway method as an acute protein depletion tool. So that pretty much summarizes what we're doing. We're now developing this TrimAway method even further. And then we're also investigating the mechanisms of TRIM21. So how is it that this ligase can degrade any substrate if it's bound by an antibody? And we're also looking at how TRIM21 functions in the body using model systems such as mice to look at how at the organism level TRIM21 can respond to things like viral infection. For many years, the lab has been looking at the role that TRIM21 has in response to various different types of viruses. And then more recently, yeah, we've been looking at the SARS-CoV-2 virus, which causes the COVID-19 disease. And yeah, we've been looking at how antibody responses to the CoV-2 virus may function via this intracellular antibody receptor TRIM21. We got this insight about 18 months ago, I think it was initially. So with respect to with the sort of standard virus infection assays we're doing in the lab, so we often use recombinant viruses that will have a GFP or an M-cherry reporter gene. And what we were normally doing was infecting cells and then leaving them a certain amount of time and then harvesting the cells and using flow cytometry to look at like percentage GFP positive cells or how intense the GFP is. And that all worked very, very nicely. And then we sort of, a colleague at the Edinburgh site had this IncuSite machine. So we gave it a try and it worked really, really nicely. So what we could do with that was do the same experiments, but now just monitor over time how infection happens and how fast it happens. So we look at the rate of GFP expression, how many cells have the GFP, and how this changes over time. And this gave us much more information because we did see with different mutations in the sort of receptor binding domains of viruses, we saw a difference in the rates of infection, but not actually in the total amount of infection and things like that. So yeah, so we've been using this IncuSite quite heavily, and it's really transformed how we've been doing these experiments. So we no longer do flow cytometry anymore. So we just use the IncuSite, and now we can do like multiple conditions, you know, on a 96-fold plate, look at all kinds of, you know, inhibitors and things like this and how they affect infection. And it's just really just multiplied like what we can do in one experiment and really saved us so many man hours. And yeah. Yeah, exactly. Yeah, we can really now just test so many different conditions in parallel in a single experiment with respect to our kind of neutralization of infection assays. And the other thing which was really great about it was just the software is like so intuitive. So anyone in the lab can just do an experiment, put it on IncuSite, push go, and then you can just then start quantifying, you know, when GFP or M-cherry starts appearing, the percentage of cells that's in, the intensity, and it's really, really intuitive. So what I liked about it was with the analysis part of the software, all you're really doing ultimately is just changing some sliders and then saying, looking at your analysis, saying, does this represent what I'm actually seeing? Then you sort of adjust it, and then you say, yep, that sort of outline of the GFP looks exactly what I'm seeing by eye, and then you just quantify all the time points and all the conditions like in one go. And it's just really been just amazing for that. And the fact that anyone, regardless of their experience, can just come in and use it and get really nice data from that. All of this was in CL1. So these are viruses that are non-replicative viruses. They don't infect humans, or they're mouse, or they're mouse viruses that don't infect humans. So yeah, our IncuSite is currently in the CL1 lab. Regarding the COV-2 work, what we've been using it for is with doing experiments with mouse coronavirus, which you can use in a CL1 lab. And it became useful there because in this case, we didn't have a recombinant virus, so we had a wild type virus, so we have no reporter genes there. But one of the phenotypes of coronavirus infection is that the cells start to fuse. So you have individual cells and then they fuse together, and then lots of cells fuse together. So you get these huge cells with loads of nuclei. And then we could use just the cell outline analysis of the IncuSite now to quantify infection by looking at how this cell fusion is happening over time. So we don't need the GFP cherry for this, but we can just use the really nice phase cell outline to quantify that. So that's how we've been using the IncuSite to look at coronavirus infection assays using the mouse model. We're going to be looking at with regards to the COV-2 virus, we're going to be looking at different, how antibodies against different components of the virus can neutralize the virus. So we've got either various different monoclonal antibodies that have been specifically raised against various components of the coronavirus. And what we're also doing is looking at serum samples from recovered patients, characterizing what antibodies they have and then putting them into our neutralization assays. So my interest was in actually this kind of protein degradation tool. And that was actually, to be honest, the main reason that we decided to get the IncuSite was because we were, so we developed this trim away method, which I talked about earlier, where you take any antibody, you deliver it to cells, and that degrades the protein that it binds to via this TRIM21 pathway. And then we're now developing new methods along these lines. And one of the kind of go-to assays that we developed was targeting GFP for degradation, right? Because then you can look at the GFP signal and you can watch it go away. So I initially did a lot of experiments with live confocal microscopy, but it's very, very tricky because you're sort of limited to the number of cells you can analyze in one experiment and the number of conditions you can do. So then I moved to kind of just endpoint assays where I was sort of doing various different kind of TRIM21 constructs and antibody constructs and seeing how they degrade GFP by simply looking sort of 5 hours later and doing a Western blot for GFP. And then, yeah, so then we sort of had a go at this IncuSite and thought it could be really useful for this because then we could get the kinetics of protein degradation, right? And initially that was quite tricky to do with the IncuSite because just by the nature of imaging through standard tissue culture dishes, you're sort of on the limit of what you can detect if you're expressing low levels of GFP, for example. Actually, I think we managed to sort of push it to its limit. So now we can express relatively low levels of GFP, various different GFP constructs that we're interested in, if they localize to the membrane, then they localize to the nucleus, and we can see those different localizations. And then on top of that, we're expressing various different TRIM21 constructs and antibody constructs that we've engineered in the lab to then target these different GFPs for degradation. So what we're now doing, is using IncuSite, expressing these constructs, and then using IncuSite to film the kinetics of GFP degradation. And like I said, I think it's really on the limit of the detection level of the IncuSite, but actually we've got really, really nice data of that. So what we're doing is we're inducing expression of TRIM21 or various, engineered constructs around TRIM21, and then monitoring immediately GFP degradation in the timescale of minutes. So we're imaging sort of max out the machine sort of every sort of, 1 or 2 minutes even, what it will allow us. I think it doesn't allow you to take up more than like 50% in the 24 hours, but we're doing these very short time courses. So we're doing like 3-hour experiments, imaging every 5 minutes and looking at degradation of GFP and then getting kinetics of half-life of GFP degradation. And that's been like really, really useful because we see differences in the rates of degradation between our various constructs. We can really easily pick up there. And yeah, like I said, with the virus experiments as well, like we can now test multiple conditions in parallel, right? And that's really important when you're looking at kinetics of degradation, because if you're doing experiments at different times, it's really hard to compare. But when you've got them on one plate, inducing everything at the same time, and then you're monitoring the rates of degradation, you can really nicely compare those conditions. So yeah, that's been really, really useful. And we actually have a paper that we're about to submit where we're showing these assays where we're looking at GFP degradation with the IncuSite. And actually, like if you look at the data, you wouldn't really you'd think it was done on a microscope, actually.